So once the lecture is over, but 9 o'clock in the morning when you have a class, and then you have to wait till, what is it, 2 o'clock before your next class? Huh? 1 o'clock. So what do you do for 3 hours? So about 6 lectures ago, I started off with structural skeletal muscle. I then went on to this idea of neuromuscular junction and excitation. I followed that through with the release of calcium and cross-bridge cycling. I then followed that through by looking at mechanisms of contraction, in other words, the mechanics, how much of force a muscle will generate and so on. Today I want to conclude, and I think this is my last lecture with you, is on energetics. In other words, where does the muscle get its ATP from? And that ATP, how is it made? So it's a kind of a blend between molecular medicine and physiology. So let's see what we can achieve with this particular lecture. So start off. This was the last slide of the previous lecture. And what the slide is looking at is if you look in the middle where it says this kind of what people used to draw, these map kind of things with computer programs in the early 80s, central nervous system. Motor cortex will then stimulate the motor neuron. Motor neuron will come to the neuromuscular junction. You get excitation, contraction, coupling, and then you get calcium signal. Eventually you'll get the muscle contracting because of the calcium, the muscle relaxing because you're pumping the calcium back into the sarcoplasmic reticulum. Okay? And this kind of flow chart thing shows you a central nervous part of it, and then you have a peripheral part of it. Peripheral meaning outside of the spinal cord, because the central nervous system is the brain and the spinal cord. And on the left-hand side, it says types of getting tired or fatigue. So obviously, the central nervous system one will be a central fatigue, and the other one, needless to say, will be peripheral fatigue. And if you look at this thing, most often than not, when we as human beings want to do exercise, we stop at psychological effects. In other words, your mind tells you 20 minutes is enough. Or your mind tells you, oh, my muscle is being injured, I'm going to stop. Or your mind tells you, tomorrow I will do 40 minutes. Okay? That's a psychological stop of exercising. Now, yes, there are people who ride the dopamine rush that they get because they are running. There are people like that as well. And they just cannot stop. They just keep running or whatever it is they're doing. Okay, so you have these two extremes. And I think most of you and myself belong to the first one. Okay? We think, that slight pain you're going to get in the ankle, oh my God, I have damaged something, I need to stop. I need to go and drink the orange juice. Okay? So that's the psychological effect, if you want. And it says nothing about the muscle. It says nothing about what's building up and so on. So let's look at neuromuscular. Again, it will take a lot and a lot and a lot of exercising for you to actually deplete acetylcholine in the motor end plate, at the neuromuscular junction. In the same token, it will take a lot for you to block off the nicotinic acetylcholine receptor. In other words, it's not available because it's too busy binding acetylcholine. Right? Then, excitation contraction coupling, obviously membrane potential. Because you require an EPP to generate an AP, okay, you require sodium going in, potassium coming out, and so on and so forth. Those membrane changes can be rectified relatively quickly. You have to be exercising extremely hard for that to have an effect. Then you have sarcoplasmic reticulum calcium. Now remember, I told you if your action potentials are coming down really, really, really fast, your circa cannot pump the calcium back into the sarcoplasmic reticulum fast enough. It's working, but it's not doing it fast enough. Okay? So SR calcium leak, or decrease in calcium release, or decrease in calcium binding to troponin C. Again, these are all, things that will take a long time for, you've got to be doing really, really extreme exercises for all of this to happen. Then you have depletion theories, and I'll come to what phosphocreatine is in a minute, PCR. ATP, you know what that is, and glycogen. Now this gets depleted relatively quickly, and I'll give you the time scale in a minute. Okay. Lastly, because of the metabolic pathways, you're going to produce metabolites. And the major metabolite is lactate, pyruvate. During anaerobic respiration, pyruvate gets converted to lactate, and lactate obviously dissociates into a hydrogen ion H+, and lactate positive, a negative salt, an ion. In other words, lactate or lactic acid will tend to cause a decrease in pH or acidic pH, and that may interfere with, tiredness and so on and so forth. So all of the ones above here, will only be under extreme conditions. Maybe this one down at the bottom here will give you something related to fatigue. And I wrote that for you on the left-hand side. Okay? The supply of ATP is obviously regulated by the amount of ATP your muscle has, which is extremely small. Okay? You can just go through, about 5 or 10 twitches with the ATP class, that's it. You build up. So there has to be metabolic pathways to give you more ATP. So you start building up these metabolites, ADP. ATP gives you ADP or AMP if you're removing one more molecule of PI. Inorganic phosphates, PI builds up, and calcium. Okay? So as ATP is hydrolyzed, you start to stimulate these metabolic pathways to make more ATP. On the other hand, ATP will inhibit these pathways during rest. In other words, you're sitting and having coffee with Dunkin' Donuts and that kind of thing. So fatigue is actually not due to a lack of ATP. It's actually due to a buildup. Again, what kind of fatigue? Remember I told you most of the fatigue is up here. Okay, I am tired fatigue, right? So it's due to the buildup of inorganic phosphates, which interferes with calcium binding or calcium release from the myosin head, lactate and H+, again, acidic environment. So that'll be a good way to now get into where we get our ATP from. Again, when the muscle contracts and shortens, that velocity of contraction, which depends upon the afterload, is actually creating work. So work, according to physics, is the load multiplied by the distance you move, whatever it is you are carrying. Okay? So ATP is required, obviously, to do that work. That chemical form of the energy is converted into a physical form of work, which in this case is moving the load. Again, what is that ATP actually used for in the muscle? It's used for these three things. The cross-bridge cycling, okay? It's needed for the myosin heads to bind to actin, because the ADP and the PI is already on the head for it to bind to the actin. It's also required for the power stroke, so you kick off the PI and the ADP, now you've got the power stroke. It's required for detaching, so when the ATP reattaches to the head, now you got detaching of the myosin head from the actin. And then you allow the cycle to keep repeating itself, provided calcium is there. Second reason you need ATP is for calcium uptake, the circle. All right? So you require calcium to pump the require ATP to pump the calcium back into the sarcoplasmic reticulum. Okay? And then obviously calcium storage is back, so when the next action potential comes along, you can release sufficient calcium. And obviously pumping it back into the sarcoplasmic reticulum will cause relaxation. The third reason you need ATP is to maintain sodium and potassium gradients, because you are creating action potentials. The sodium is going in, the potassium is going out. You need the sodium, potassium, ATPase to pump that sodium out again and the potassium back in again. Okay? Where do we get the ATP? So ATP provides the energy for the contraction. So if you're going to have sustained contraction, you require lots and lots of ATP. Okay? We have a store of ATP, or sources that will give you ATP. The amount of ATP is minimal. Okay? It's absolutely tiny. Again, muscle fibers must synthesize more ATP if you want that muscle to contract. Again, we have ATP, which again, absolutely minimal. I just want to find a number here. Can you see ATP in the table? Yes? Okay. It says the number of twitches about 10. All right? How quickly will you finish using it? Within 2 seconds. So it's a total waste of time in terms of actual ATP. Okay? So, ATP, you have ATP, then you can convert the ADP, so the ATP gives you ADP plus PI, you can convert the ADP by using a storage form of phosphate, a high energy phosphate obviously. It's called creatine phosphate. So you transfer the phosphate from the creatine to the ADP, and then the ADP becomes ATP. You need an enzyme for that called creatine kinase, and you've come across this enzyme when you did clinical enzymology in molecular medicine. Okay? This is that enzyme that tells you whether or not the muscle is breaking down. If it's skeletal muscle, it's called rhabdomyolysis. This is the enzyme that tells you whether or not somebody has a heart attack. CKMB. You know these words, yes? Yes. All right? And this is the enzyme that tells you if somebody's got a problem like polymyositis. In other words, an inflammation of the muscle. Okay? So creatine kinase converts creatine phosphate plus ADP into creatine plus ATP. And now you can use the ATP. However, even this is limited. If ATP is 2 seconds, this is like 10 seconds. The creatine phosphate. Then what happens? Now you go into biochemical pathways. Pathway number 1, anaerobic glycolysis. So you've got glucose either in the blood that's supplied to the muscle or a little bit in the muscle. as glucose 6-phosphate. Okay. So where is glycolysis taking place, or anaerobic glycolysis? Obviously in the cytoplasm. You do not require oxygen for the reactions to take place, but it generally takes place when oxygen is not available. Okay? Again, it is the main source of energy when you require ATP very, very, very quickly. In other words, high frequency stimulation of glycolytic fibers requiring a lot and a lot of energy for the muscle contraction. Jumping, running, that kind of thing. How long does it last? It lasts anything from 30 seconds to about 2 minutes. Okay? In other words, the minute your glucose is low, that's not because the glucose in your blood is low. The glucose in the muscle cell is low, or the glycogen is used up in the muscle cell. Now, this process is not going to give you sufficient ATP. Again, you are using glucose and stored glycogen. Okay, ATP yield can be very, very fast in these reactions, partly because it's in the cytoplasm. In other words, it's in the right place kind of thing, where you need that ATP for the muscle to contract. Obviously, there's a net gain of 2 ATPs and then a pyruvate, all right? Again, when the oxygen is limited, you need the NADH that's formed in the reaction. So what you do is you convert the pyruvate to lactate, and now you use the NAD that's formed to keep the anaerobic glycolysis going. This decrease in pH because of the lactate is what gives you the supposed, the muscle burn, if you want, when you're exercising. Okay, then what happens? Well, now you have oxidative phosphorylation. Please, I want you to understand something. Even though I'm presenting this from the least amount to like the greatest amount of ATP, that does not mean the muscle uses it in that fashion. Everything is working kind of all together here, okay? Okay, where is this thing taking place? Mitochondria. Again, you must have oxygen, because the oxidative phosphorylation, the cytochromes do not work if there's no oxygen. Again, the ATP production can be prolonged. Again, think about a marathon runner running for, apparently the guy did it under 2 hours the other day somewhere. Which one was that? London, whatever? Yeah?
Yeah, he did it under 2 hours.
Yeah.
His name?
What?
His name is Eliud Kipchoge.
Yes.
No.
Yes, last week I think it was on the news. So think about the marathon runner. For you, it's like 5K. I do not know if you guys do 5K. So you can generate this energy for a very, very prolonged time. Okay. Again, the muscle activity cannot be maximal. It's got to be sub-maximal. The fuel source obviously is glucose, but you can also use fatty acids. Okay, so excuse me. You can use pyruvate from anaerobic glycolysis. You can use pyruvate. You can use fatty acids. The pyruvate, by the way, gets converted to lactate. Lactate now goes to the liver and gets reconverted to pyruvate and glucose and comes back to the muscle. You know that as Cori's cycle, C-O-R-I. Okay? You can use fatty acids, which the muscle does that. And you can also use amino acids. That doesn't mean you break down the proteins. You're just using residual alanine and so on and so forth. Ketogenic and glycogenic amino acids that might be around. ATP yield, because there's so many reactions here, tends to be slow, but it's slow to activate, but very high efficiency. Depending upon who speaks to you about this thing, they can tell you 36 to 38. This thing says 30 to 32 because you are using the ATP in the glycolytic pathway and so on and so forth. ATPs. Okay? Again, it is for sustained submaximal stimuli or contraction of the muscle. You can use fatty acids, 20 times more ATP than obviously glycolysis. glycolysis is just tube. Okay? In glycolysis, the end product is pyruvate, which eventually gets converted to lactate. Here, the end product is carbon dioxide and O2, and now you need to get rid of that through your respiratory system or through the kidney. Okay, whichever way works here. I summarized that for you in the table. Please do not send me an email saying, do I need to know 320, 100, 100? Do I need to know 1070, 670, 12,000? Do you think you need to know that?
No.
No. Don't use your precious neurons for stupid things. What do you need to know? Very little energy, 2 seconds. Just need to know very little energy for contraction. Very little energy, at the very most about 10 seconds for creatine phosphate. That's all you need to know. About 130 seconds. Obviously, if you need to open another textbook, okay, you'll find a slightly different number here for 130. And so on. So about 40 minutes. Now, you might say, I just told you that glycogen with aerobic respiration can go on for 2 hours. Yes, I told you that. But here it says glycogen. It doesn't say glycogen plus fatty acid plus glucose plus protein. No, it says glycogen. And only glycogen is around 40 minutes, which is a nice number, because if you're trying to lose fat in exercising, submaximal exercising, just running on treadmill lightly, you must do it for greater than 40 minutes, because that's when you start to lose the fat or break down the fat. Okay? Okay. So are we okay with this table? And I gave you the reactions, not summary reactions. I then summarized that for you in a table. Again, about 15 seconds for phosphocreatine. Okay, creatine kinase will transfer that phosphate, that high energy phosphate, from CP to ADP. Again, the thing to know is it is an immediate backup for ATP when you start exercising, obviously. Elevated creatine kinase is a marker for something that's breaking down. Because we are doing MSK, it will be skeletal muscle, and that's called rhabdomyolysis. Obviously, if you're doing the heart, then it is cardiac muscle, in which case it's myocardial infarction. Okay? Anaerobic glycolysis, again, breakdown of glycogen and glucose inside the cell. You get the energy very, very fast, high intensity exercise, but very inefficient, obviously, all right? And again, lactate buildup will cause low pH, and this generally is what results in delayed onset muscle soreness. Okay? That soreness that you get, you know what is soreness? The pain. Okay, that pain. That soreness that you get the next day. The one you get is actually due to like myofibril damage and so on and so forth, which is actually a good thing. You're doing bone presently with Dr. Khalid. Having micro fractures, not fractures, micro fractures in the bone is a good thing, because it allows the osteoblasts and osteoclasts to recycle that particular weakened part of the bone. The same principle here. That soreness that you get the day after is actually those myofibrils are kind of a little bit torn kind of thing. Then your body repairs it. Lastly, aerobic oxidative phosphorylation, again, anything greater than 2 minutes up to 2 hours. Okay? Again, pyruvate fatty acids and amino acids through the TCA cycle, and oxidative phosphorylation gives you tons of ATP. Predominant source of ATP for long distance runners. I then put that, sorry I'm repeating myself, I put that in a diagram for those of you who like diagrams. So what is the source of energy when we are at rest? All of us right now. I'm working a little bit because my postural muscles are working. I'm trying to use the wall. Okay? So, there we go. You have a blood vessel, and then remember, glycolytic fibers have a high supply of capillaries, they have a high supply of myoglobin, and therefore they have a high supply of stored oxygen. Okay, there's a blood vessel, there's a excuse me, a myofibro, and you can see the actin and myosin inside. So, at rest, what's happening? You're generally breaking down fatty acids. You're getting some glucose, you know, from the Dunkin' Donuts that's getting into your muscle cells as well. Because you're not using the muscle, that's generally is used to store into glycogen. Obviously, that is filled up. Khalas, that's filled up. All right? What happens to the fatty acids? It comes in, goes into the mitochondria, gets converted into ATP, beta oxidation. What do you do with the ATP? You then store it as creatine phosphate. Again, at rest. So you're building up the stores of glycogen, you're building up your stores of creatine phosphate. Are we okay so far? What happens if you keep putting in the fats? You start to build up the fat around the myofibro. Okay? As fat, triglycerides. Okay. So, in a resting muscle, the demand for ATP is extremely low. You're not bringing about any contraction and cross-rate cycling. Resting muscle will then use fatty acids, glucose, and so on. Some mitochondrial ATP is converted, okay, to storing it as glycogen. Some are used to build up the stores of creatine phosphate, and those are obviously restricted. It's not like it just keeps making that and keeping it. So the result is essentially to build up all of these sources of ATP, so you need it, okay, when you decide to either walk or run behind your friend or whatever it is in the corridor. So, what happens in moderate muscle contraction? Again, in moderate muscle contraction, you're still using fatty acids that come in, you have sufficient oxygen available, you use the glycogen. Now the arrow is the other way, the glycogen is giving you the glucose is giving you pyruvate, you've got some ATP there, you're getting, because there's sufficient oxygen, you get sufficient ATP from oxidative phosphorylation, and all of that is going towards muscle contraction. So this is submaximal contraction of the muscle with the availability of oxygen and fatty acids. So during moderate levels of activity, your ATP is essentially coming from oxidative phosphorylation. There's still enough oxygen around to do oxidative phosphorylation or carry out oxidative phosphorylation. The muscle relies primarily on aerobic respiration. If the glycogen reserves are low, okay, then the muscle can also break down other substrates like fatty acids. So you're going from the 2 second to the 15 or the 10 seconds upwards. If all of the ATP is used, all of the ATP now produced is used for muscle contraction. So it's not like the contraction is still going on, but there's no ATP. Now that would be in the next slide. Okay? Now you got peak activity. And in this case, you got a blood vessel. Notice, notice it's not showing you the fatty acid coming in or the oxygen coming in. Because you're going quickly into this thing and you want the maximum contraction. So what's going to happen now? Glycogen is going to give you glucose, glucose is going to give you pyruvate, you get the 2 ATPs, that goes straight into contraction. Creatine phosphate will convert ADP into ATP, that goes straight into cross-bridge cycling. Okay? So where's the mitochondria here in this particular example? The only reason it's not shown, yes, it's generating some energy. The only reason it's not shown is because under peak activity, the oxygen supply is less, and therefore that source of energy is only available for as long as the oxygen is available, oxidative phosphorylation. So, during peak activity, the demand for ATP is very, very great. Okay, I told you yesterday, this is your trainer. Even though you've been running for, I don't know, 20 minutes on the treadmill, he comes by and he tells you, I want you to run fast as you possibly can. I want you to turn up the speed for 2 minutes. Now you are in this thing. In other words, in that 20 minutes where you were kind of just running, you were doing this. Are we okay? So in that last 2 minutes, he wants you to do this. Are we okay? Good. So you need to meet that contraction. Put the hand down, I'll take it in a second. So only 1/3 of the ATP's need will be met through the mitochondria, but it's not shown here as a mitochondria like it was shown in the previous slide. So this is peak activity. Now you know, even though we all don't go to the gym, you don't go to the gym and start off immediately on peak activity. Okay? All right. So where is the ATP coming from? It's coming from anaerobic glycolysis. Obviously this pathway here. Where else is it coming from? It's coming from the utilization of creatine phosphate stores. Again, pyruvate is converted to lactate. Lactate will now be pushed out of the muscle into the bloodstream. It'll go to the liver, be reconverted to pyruvate and some glucose, and then sent back to the muscle. But you're also building up hydrogen ions, so you're going to get, in peak activity, you're going to get quick fatigue of the muscle. I wrote that for you in this slide. Cori cycle, removal of the lactic acid to the liver, conversion back to glucose, and then sent to the muscle again. Now, in peak activity, because the oxygen supply in the muscle, not in the body, in the muscle, okay? Dr. Hashim told you oxygen supply goes from approximately 98% to about 75, then goes back to 98. It's not like your blood does not have enough oxygen. Your muscle does not have enough oxygen. Are we okay with that? Yes? Good. Okay. So, oxygen debt. So when you exercise at peak, that last 2 minutes or 5 minutes, the guy told you to run, and then you start panting. What are you doing? That's not because you have less oxygen in your blood. Why are you panting?
Have more CO2. Have more
You
acidity.
are trying to get that oxygen into the muscle to get rid of that hydrogen ion, that carbon dioxide. That's what you are trying to do. So, after exercise, the exertion, that panting, the body needs more oxygen to normalize those metabolites. The lactic acid, the lactate, the hydrogen ion, the carbon dioxide, those things, okay? Resulting in the heavy breathing. This is what we call excess post-exercise oxygen consumption. Okay? Obviously, all of this is terribly inefficient because every time you make ATP, you are losing energy. Okay? So you're generating heat. So, active muscles will produce heat. About 70% of the energy that is produced can be lost as heat. I am not finished, but I'll finish this lecture. Okay? I'll take the question.
Doctor, when we said we switch from low to moderate to peak activity,
Here.
is this, yeah, so all three phases, are we also switching between the bits of muscle fibers, like the fast?
Thank you. Thank you for bringing this up, because I forgot to mention that. It was in my mind as I was talking. Okay? What is being pointed out to me is you have these three varieties or types of muscle: the slow twitch, the intermediate twitch, and the fast twitch. Okay, so effectively what you are doing is, this thing is kind of describing the metabolic pathways of those twitch kind of muscles. So if I look at this slide, since I'm on it, this will be the predominant metabolic pathway for which twitch? The intermediate and the slow twitch, because it's oxidative phosphorylation. Does it mean that this muscle cannot use glycolysis? No, it doesn't mean that. It's got a lot of blood vessels, it's got a lot of myoglobin, oxygen that is, it's got a lot of mitochondria. So its predominant pathway is this. Can it use glycolysis? Yes, it can. Does it have creatine phosphate? Yes, it has. On the other hand, if you look at my friend, the large one, the third one, the fast glycolytic one, its predominant pathway is this. Okay? But does it have mitochondria? Yes, it does. It's not a red blood cell. Okay? It has. But the proportion of the ATP that will come from that mitochondria is lower. Therefore, the amount of time that the muscle will contract using this metabolic pathway will be very, very small. Okay? So, to come back to the answer to the question, can the other pathways work in those different muscles? Yes, they can. However, it is your brain, through the frequency of stimulation, that will bring in those contractions of those other types of muscle fibers. Okay? Good. Yes, Tariq. Let me switch slide presentation style. You can talk, Tariq.
So we said during moderate activity, the large, or the CKMB and the anaerobic glycolysis can happen also.
Yes.
Which happens in the slow, fast muscle.
Yes.
But we said that the fast
They,
muscle-
they all happen in all muscles, all muscle types. Okay? But the predominant one for the slow ones will be oxidative. Okay? Yes, go ahead.
But we said during moderate activity, the fast one cannot be recruited. So how will they make energy without being-
Which is what I said at the end. I said at the end, so if you take me for instance, I'm standing, I got postural muscles. I started off with the slow ones in my muscle. My brain started off with the slow ones in the muscle. Then might have switched to one or two glycolytic ones, then switch back to some different slow ones. In the meantime, the ones that contracted first are relaxing. They're rebuilding their CP, their glycogen, and so on and so forth. Okay? So it's the brain that's doing that for me right now. Now, there is a point you are raising here. I'll come to it once I finish this. Okay? You are going to come to the lab next week. There is a lab. I will send out an email the minute I'll put the presentation and the lecture presentation, this presentation, and the lab schedule of when you individually should come to the lab. Okay, so M1.1, 1.2, 1.3 will come 9 o'clock someday, whatever it is. Are we okay? And I'll put that, I'll send it to you personally to your email. I will also send, like we did with ECG and PFT, that lab generated handout, you know, that thing you need to read. This time it is not 18 pages. I don't know. It's about 6, I think. The first 2 are introduction and then the rest, whatever. Please read it. So, you are going to do this thing called electro, which comes from electricity or excitation. myogram. In other words, you are going to measure the excitation of your friend's muscle. It's called electromyogram. This does not require you to stimulate the muscle. Because we are going to use a voluntary muscle, all you need to tell your friend is flex the arm. And then the muscle will contract. Are we okay? And we are going to try and measure that electricity. Yes? Good. So, again, EMG is recorded, is the electrical activity in the muscle. How do you do this? You place 2 electrodes, a positive and a negative electrode, on the belly of the muscle. So if you're doing it on the bicep, you keep it about 5 centimeters apart. You put 2 electrodes. You know how that machine works. It tells you put green here and red there and all that story. Yes? Okay. And then you tell your friend, flex the bicep. Obviously, my 5 lectures now will tell you, okay, the action potential will come down, the muscle will create, and now you do that, you should record something. Why would you as a doctor need to do this? Now you won't do it. The person in the lab will do it. Or if you are a neurologist, if you decide to become, you do neurology, not urology. Urology is bladder. Neurology, then you might be in the room while they do this. The nurse sets the thing up, you come in and do this. Why would you want to do this? You want to find out whether that patient's weakness, muscle weakness, is due to a muscle problem or not necessarily brain. The conclusion would be maybe brain. It could be spinal cord. So it could be a neural problem, a nerve problem, a neuropathic problem. Okay? Okay. In a disease called amyotrophic lateral sclerosis, you have to do this to figure out this motor neuron that would be damaged. It's the central motor neuron that's damaged. Okay? So this is why you do this in a lab. Or you will do like the play version of it in the lab next week. Again, just to represent that, you got the spinal cord, the brain, you got the motor neuron or the ventral horn coming to the muscle here. You then blow up that muscle, there we are. It innervates that one motor neuron or alpha motor neuron, innervates all of these fibers, and the fibers are now shown in darker colors here. Notice that that one motor unit for that motor neuron is distributed in the entire muscle. Yes? Yeah? Now, that should make sense, because if that motor neuron only came and innervated the muscle here, when that muscle contracts, the other part will not be contracting. That muscle will not be able to make this bone move or whatever it is. So therefore it's distributed throughout the muscle. And then you have other motor units as well. So again, that's what's happening here. You're going to stimulate this thing, or this is what you're going to measure. Again, how do you represent this? There we are. We have motor units, green ones, we have blue ones. Again, if you stimulate the blue one, because there are lesser fibers attached to it, you get a small amount of maximum contraction. The green one, if you do both, then you get a maximum contraction up here on the top. So you can actually recruit a motor unit. So in the lab, you tell your friend flex the muscle. You will get a recording, you'll see what it looks like. You then put a weight, a dumbbell. We'll give you a small dumbbell. Okay? And then your friend does that, give you another dumbbell. You do that. In other words, you're going to do what? You're going to recruit more motor units. Now those of you with well-formed biceps, you will not find any changes in the thing. But those of you who are like me, you will find lots of changes between the small dumbbell and the middle dumbbell and so on and so forth. And this is how you set your friend up. They come in, you put these 2 electrodes here, you put the 2 electrodes, we'll also do this, we'll look at the extension of the arm on the tricep. It just tells you which one to do and so on and so forth here. The machine will all be set up for you. Are we okay? Good. This is what it would look like. So, if you are looking at this, it says bicep. Can you see bicep down here? Yes? Can you see these spikes? Notice the bicep is quiet, it's quiet. And then your friend will do the flexion. And then you have these spikes. Now there's the spikes blown up. What are these spikes? These spikes are the voltages. Now obviously, the machine does not know it must only take the voltage for depolarization, or the voltage for EPP, or the voltage for muscle action potential. You understand? It's just taking everything. Any electrical activity. So now what must we do? Not we don't do it, the machine does it. The first thing it does is it takes out all the noise. The second thing it does is, if you look at the spikes, if I draw a line here at 0 across, I got some that are negative and some that are positive. What the machine does is it takes all the voltages and it makes them positive. So any repolarization that's going in a different direction or whatever it is, it makes it a positive value. And then it gives you this one on the top. So these are the spikes. And then it says here, can you see INT bicep? It says an integrated reading of the bicep, which comes from this. So you're looking at this, and then you got that. Now that's telling you that that bicep is contracted. Are we okay? What's the blue line, and I do not know what color is here? The tricep. And that's what you will see. It's a simple experiment. I'll tell you to do different things, the one with the dumbbell and all that nonsense, okay? And I'll also try, you must try and do this. You need to get an interference pattern. Now, what is an interference pattern? This is what Tariq was asking me earlier on. If you got a sequence of stimulation of slow, intermediate, and big fibers, all right? Now, the brain does that, but how do I make them fire all at once? How do I turn it into kapsa, if you want? And that's what you need to do. How do you do that? You create a strong isometric contraction. Are you with me? Like I did when I told you lift. Remember I was trying to lift? Now that's a strong isometric contraction. What happens there is you do not follow the pattern. And that's what's going to happen here. It's called an interference pattern. Now when you do this, it tells you something. And let me explain to you very quickly in 1 minute. The top one is telling you normal interference pattern. In other words, what you are measuring is this green thing. Okay? Now if you look, it has a voltage on the Y and it has a time scale on the X. In a person who has a problem with a motor neuron, it will look like the B. In a person who has a problem with the muscle, notice the frequency has not changed, but what has happened to the amplitude? It has decreased. So this is a myopathic problem. It tells you here, polymyositis, whereas B is a neuropathic problem, a muscular atrophy, or a neuropathic problem. And that's how you use this thing. I will also ask you to do, when you do a flexion, generally your triceps should be relaxed. Yes? When you do an extension, your tricep contracts, but your bicep should. And I'll ask you to do that as well. I'll show you how to do that in the lab. Now I am done. So, when I send you the email, please read the papers, and I'll see you next week.
Thank you very much.